So let's imagine that there's a gene expressed only in the cell that's indicated by the arrow below. What can you predict about the structure of that gene? In order to solve these kinds of problems, you need to think the transcription factors it has, HOX2, dorsal, and segment anterior. Only if a gene has binding sites for HOX2, dorsal, and segment anterior will we get. So in this particular cell, this gene would be expressed, but it would be expressed nowhere else because no other cell has this combination. This one has HOX2 and dorsal, but it's segment posterior, the wrong one. This one has HOX2 and segment anterior, but it has ventral, not dorsal. So only in this cell will we get, let's call this gene X, so protein X, only in this cell will we get X and nowhere else. So similarly, you can decide if a particular gene induces some structure. In animals, the gene Tin Man induces heart formation. The Tin Man lacked a heart, and mutations of this gene produce fruit fly embryos that lack a heart. And so the gene Tin Man, you can predict it should be expressed somewhere in the cells at some stage in development. And so depending on what you know, if you know that hearts tend to be somewhat dorsal structures in insects, and so if this is an insect, you might predict that Tin Man is here. On an exam question, you could equally decide that Tin Man was in some other location. And then once you have a location for it, design for Tin Man the transcription factor binding sites that would result in Tin Man being expressed in that cell and no others. And you could do the same thing for, in this case, distalis. And in this case, you'd put it in more than one location because limbs appear in more than one location of animals. Distalis regulates limbs, wings, fins, depending on the organism.